Selamat siang, Bapak Ibu. Sebelum kita mulai, apakah suara saya terdengar? Ya,
Terdengar.
oke. Baik. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Terima kasih. Thank you for joining us today. Kita akan mulai acara webinar hari ini akan diagendakan yang diagendakan oleh NBO Indonesia. Oke. My name is Aida. I'm going to be MC today, and Mr. Handy going to moderator today. Will be going to be moderator today. Baik, Bapak Ibu. Oke, pembicara kita sudah join. Bapak Ibu di guide ini saya ingin menyapa dulu. Greet our speaker, Miss Rusini. Are you with us?
Hello, hi.
Hello, hi.
Hi.
How are you?
I'm fine, thank you. Selamat datang.
Yeah, selamat datang, guys. You have joined it already, yes.
Sorry, what was that?
Miss Rusini tadi sudah join jam 1, Bapak Ibu ya tadi. Lebih
ya,
cepat, yes.
Saya try dulu.
Sorry, yeah. Malaysia, yeah, is 1 hour ahead of Jakarta, yes, yeah.
Yeah, it's earlier for us.
Yes.
Yeah.
Yes, oke. Baik, Bapak Ibu, sebelum saya memulai acara hari ini, marilah kita berdoa bersama-sama agar acara kita berjalan dengan lancar. Berdoa dimulai. Berdoa selesai, thank you. Oke, before we move to first presentation session, going to share about the next agenda in NB Indonesia, Bapak Ibu. Jadi NB Indonesia punya acara-acara berikutnya ya setelah webinar ini, dan saya akan share apa saja agenda-agenda berikutnya sebelum sesi presentasi ini yang akan dipandu oleh Pak Hendy akan saya serahkan ke beliau. Baik, saya akan izin share screen dulu. Oke. Oke, Bapak Ibu, terima kasih sekali lagi telah bergabung di acara kita pada hari ini, dan our speaker luar biasa is Miss Rusini here ya with us. And ini adalah next agenda di Indonesia yang ingin kami sampaikan. Jikalau Bapak Ibu berminat dan tertarik untuk join, silahkan daftar sekarang juga. Nanti juga ada ini network farmakologi ya Bapak Ibu ya di November dengan pemateri Bapak Didi Husotomo PhD, founder of InBio Indonesia dan juga tim di InBio Indonesia, dan juga ada materi tentang optimizing Grammarly Bapak Ibu ya. Nanti kalau berminat bisa langsung daftar juga. Dan ada consulting your research juga, dan BRC InBio juga Bapak Ibu ya. Ya ini free juga. Jadi banyak tersedia next agenda di InBio ini yang free maupun yang berbayar juga Bapak Ibu ya. Nah ini juga ada International Conference Bapak Ibu ya, kerjasama juga InBio dengan Ecobios ya penyelenggaranya, dan juga ada di InBio ini private training program Bapak Ibu. Jadi jika sekali lagi berminat daftar ini, monggo bisa chat admin kami. Nah, ada BRC InBio juga, dan call for ebook publishing juga Bapak Ibu. Dan ini beberapa poinnya juga ada yang free, bisa langsung menghubungi tim kami juga. Dan tentunya tidak hanya program, ada juga tools yang support untuk penelitian bioinformatik. Di sini juga ada kita ya secara dynamic, dan juga tools lainnya ya Pak Ibu ya. Jika tertarik silahkan juga menghubungi admin kami. Dan nantikan acara-acara berikutnya ini akan selalu kami update Bapak Ibu ya di platform Jari InBio, di Instagram maupun di website kami. Oke, baik, itu saja yang ingin saya sampaikan dari acara-acara yang akan diselenggarakan oleh NBU di next session-nya Bapak Ibu ya. Baik, kita akan memulai acara pada hari ini. Pembicara kita, our speaker, Ms. Rushini, akan menyampaikan materi tentang topik kita hari ini, dan yang memandu acara pada hari ini ya bukan saya ya, ini spesial banget juga. Ms. Rushini dari Evendop, dan yang mandunya adalah Pak Hendy ya. Kepada Pak Hendy saya persilahkan.
Oke, ya terima kasih Bu Aida atas
Ya.
introduction-nya, dan terima kasih juga dari tim InBio Indonesia, ada Mbak Evi, ada tim Mbak Ruri dan tim ya. Dan ini memang webinar kita kerjasama kedua ya. Pertama waktu itu kita sempat bahas tentang pipetting pendok, dan kali ini kita ada kesempatan untuk berbicara tentang penelitian sel punca ya, atau stem cell yang memang Miss Rusheni Munis Parandes ini sudah ahlinya lah di situ ya, karena memang Ibu Rusheni ini mungkin profil singkatnya ya sebelum kita masuk ke sesinya, memang dari University of Putra Malaysia ya, dengan master degree in molecular medicine, and beliau join di Evendop tahun 2020 sebagai technical product specialist, dan memang provide application support, lalu juga ada troubleshooting, theoretical, termasuk nanti challenges dan workflow dan solution juga tentang penelitian stem cell ya, untuk kesempatan siang hari ini. Dan mungkin nggak berlama-lama, Hai Rusheni, are you ready for delivering your presentation, and the time is yours. Yeah.
Okay, thank you so much, Handy. Thank you so much, Aida, for the introduction.
Yes.
Yeah. Okay, thank you so much for everyone for joining us. I can understand Bahasa Indonesia a little bit.
Wow.
So it's a little bit similar to Bahasa Malaysia, I think.
Yes,
so,
Melayu.
Yeah, Bahasa Melayu. Yeah, that's right. Yeah. So, but today I will conduct the session in English. But feel free to ask questions. Anything in any way you feel comfortable. You can use the chat, or you can just unmute yourself. I think that's also okay. We can have a very a discussion session. Yeah. Okay. So I wanted to say good afternoon, because I know it's a little bit later in Indonesia now, so maybe about 3:30. Yeah, so thank you for joining us for this talk. So maybe I'll just introduce myself. But Handy did a good job introducing already. My name is Rushini. I'm part of the technical product support team in Kuala Lumpur, working on regional support, application, and also product support for Appendorf. So today, I will share with you a little bit on stem cell derived exosomes. Let me share my screen.
Yes.
Okay, so you can see my screen?
Yes, you did. Yes.
Okay, great. Okay, so today I will share with you a little bit on stem cell derived exosomes. some of its challenges, especially to culture it, and also hopefully what is the enough solutions for these challenges. So without further ado, let's get started. So I want to start with what is stem cells? So actually there are many different types of stem cells. Maybe can I? So, okay, let me just use this pen to show you the different types of stem cells. For example, there are embryonic stem cells, and all of these different stem cells are categorized in different classes. So embryonic stem cells are considered totipotent, and that means they actually can differentiate into any cell type. So embryonic stem cell, an example is actually the zygote. So the zygote is only a single cell, but it can differentiate into every cell in the human body, your hair, your teeth, it's all very different, but it has the ability to differentiate into all the different cells just from one cell. And then we also have embryonic stem cell, or iPSC, which is induced pluripotent stem cell, and they are categorized in the class of pluripotent. That means they can differentiate into a few different types of the 3 embryonic germ layers, which is the endoderm, mesoderm, and the ectoderm. So they have a little bit less ability to differentiate than totipotent, but it's still quite a lot. Then we have the more common adult stem cell, like mesenchymal stem cell, hematopoietic stem cell. Okay, which has the class of, is considered multipotent because they can differentiate into a few different cell types, but it's a bit limited. We also have adult stem cells that is considered only unipotent, so they only can differentiate into a single cell type, like epidermal cells. So we have many different cell types here. Okay, let me stop this. Okay. Okay. So, and unfortunately, I don't think we can cover all of these cell types. So today we will concentrate on one type of cells, which is quite common, I think, used in clinical trials and research across Southeast Asia and globally. And that is actually the mesenchymal stem cells. So the mesenchymal stem cells in the use for therapeutics has a very wide range of use. They can be used to be differentiated for hair reconstitution. They also can be used for transplantation for liver, especially in cases of liver failure or bladder tissue. They can be regenerated into muscle cells in the case of muscle degenerative diseases. They can even be regenerated to repair heart scars, mainly from a heart attack. And so there has a lot of therapeutic potential. So since we have so much of therapeutic potential, maybe you're wondering, where does it start from? So actually, mesenchymal stem cell has a very long history. So I know it feels like 40 years, 1960, but actually it's 60 years already. Yeah, so we are in 2023. So MSC has a 60 years of history. And I think in 1991, it was finally defined as mesenchymal stem cells. And then in 2001, we have some reports on some characteristics that make it mesenchymal stem cells. And in 2006, we finally have the full characteristics. For example, it was finally defined. So this category of cells is considered mesenchymal stem cells. So it has to fulfill a certain characteristics. We have presently many papers looking at stem cells and even clinical trials. So if you go online to clinicaltrials.gov, actually you'll find many clinical trials that actually currently working on mesenchymal stem cells. So mesenchymal stem cells, okay. So mesenchymal stem cells is not new. So it has 60 years of history. So maybe you're wondering, what is the market potential? So actually the market potential is very big for mesenchymal stem cells. In 2023, it's about globally. So this is about 12 billion USD, and it is expected to rise. And in 2030, the market potential for stem cell research and therapy is expected to be about 31 billion USD. So it's a very big potential there, and very long history as well. And I think that's maybe why you're also in this call today, so that we also want to join and see how can we harness this potential. Okay, so in order to talk to you about the challenges and some of the solutions that we can give, first I need to talk about the workflow of mesenchymal stem cell. So I have here a general workflow of the mesenchymal stem cells. So it first starts with a sample collection. So here it's a primary sample. So I have an example here of one of the ways to get mesenchymal stem cells. It is using the baby's umbilical cord cells. So from here, they can derive mesenchymal stem cells. So when the baby is born, you can actually harvest the umbilical cord to get the mesenchymal stem cells. So this is one of the ways to get the primary sample. After that, you see this sample is very big. So we need to process it, make it smaller. So you need to mince it. So there's a mince, they will do some sonification. So there's a processing steps involved here. And these processing steps, you will need to use centrifugation and you have to prepare for culture. So when you have single cells, then you can actually cell culture them using T-flask, cell culture discs. And of course, you also need to put them to allow them to propagate. You need to put them in a CO2 incubator. Minimum, you need CO2 incubator and at least 5% carbon dioxide concentration. And I'm pretty sure many of you here are familiar with stem cell culture. So one thing that you have to do when you do stem cell culture is you have to visual check and you have to do maintenance or passage every single day. They're very sensitive, so they need lots of, when I work in the lab, we call it tender love and caring for the stem cells. Every day you must give them love. So you must check every day, are they doing okay? And if not, you have to passage them. When they're confluent, you need to passage them. So we call it harvesting. So when we harvest, we also use a lot of liquid handling tools and serological pipettes. And you need to do some centrifugation for process. And here's downstream analysis. So here's a little bit open, depending on what is your experiment. You want to analyze the stem cells, you want to use them for therapy. And then final step is once you are done with your experiment, you need to store them. So you need to store them for a short term in -80 and for a longer term in liquid nitrogen. So that is the workflow for stem cell culture. So I like to divide this workflow into a few parts. So we look into each of these parts, what is the challenge and how we can have a better solution. So the first part, the sample collection part and processing of this sample. So what kind of challenge do you face at this very beginning stage? So one of the main challenge here is the low yield. So although this sample looks very big, but actually once you process it, the amount of mesenchymal stem cells that you can get can be very, very, very low yield. So that is a challenge in primary stem cell culture. And second thing is what we call shear stress. So it's a kind of stress that is implied on the stem cells during the processing step. So when you are doing, when you're using the liquid handling tools to mince the sample, to process them and using centrifugation. So these steps can introduce what we call as shear stress. So we'll look into that a little bit deeper later. And last but not least, and maybe what we all experience is very tedious and very time consuming. If you work with a stem cell, they can take a very, very long time. So this is some of the challenges. So this challenge is very common, but what is the implication if you do have this challenge? Let's say your stem cell has very low yield. So of course, the main thing is your downstream experimentation is affected. If you don't have enough cells to propagate, you may not be able to proceed for sufficient downstream experiment. And if you're looking to create a way to finally use this for therapeutics. So you're in R&D, want to scale up to preclinical stage, clinical stage. So scale up is going to be very difficult. And normally, traditionally, we use a culture flask or T-flask to culture stem cells or any cells as well. And the thing about T-flask is there's no control. You have no idea what's happening inside the media. And also it's very hard to scale up. And well, yes, this is true, but I must support with evidence. So actually there's a lot of papers just on the internet that you can find that talks about that the fact that actually MSC culture of umbilical cord has very low yield and it's very limited by the types of cultural method as well. So just these screenshots are just a few examples. There are many, many papers. I just took a few to show you that there are many people who actually also experience these kind of challenges. So we have two challenges. One is low yield and another one is shear stress. So before I want to talk about shear stress, I want to hear from you. So actually what you can do is you can just type in the answer in the chat box. You can say yes or no. I just want to know, there's no correct answer. You can choose anyone. If you are familiar with shear stress in stem cells. So you can just type in the chat box.
Which one, guys? Yes or no? No.
No, okay.
No.
Okay.
I'm no too.
Okay. Okay, that's I mean, that's good. Then we can all find out together. So I think it's a pretty new, not so new, but a little bit newer topic.
Okay.
Generally, shear stress. So you can actually maybe after today's session you can Google it to find out more. It's very interesting. I think shear stress, something new that we are discovering. So I see most of the answer is no. So I will give you a little bit of just a very, very brief background on shear stress. Okay, so shear stress is a stress that is, so it's like stress that is applied to the cell when you're processing it, when you're using the pipette to aspirate, resuspend the cell, when you're using centrifugation to spin down the cell to throw away the supernatant, when you're doing mechanical mincing of the cells, sonification from, for example, from the umbilical cord to a single cell. So all of this process, everything you put through, everything the cell experience is very stressful for the cell. But this stress actually can cause something we call shear. So shear actually directly translates to like tear. So the cells can tear. Yeah. So they can actually like break apart. So but so this can happen to any cells, not just stem cells, normal cell culture, even cancer cell, but cancer cell is a little bit more robust. So different types of cell, it can happen. But it is more prominent in stem cell because stem cell is very sensitive. So it's a little bit more stronger in stem cells. So it does not just, sometimes it can shear the cell, the stress applied. That means the cell actually break apart. So that means, of course, the cell is already apoptized and is no longer, cannot propagate anymore. But sometimes shear stress, when applied to the cell, especially stem cell, does not only cause it to tear apart, shear, but sometimes it can cause it to lose its stemness characteristics. So like just now, we looked at the timeline. In 2006, the Society of Stem Cell decided what is the criteria for a cell, for a group of cell to be called stem cell. So that cell have to have those criteria. So when it does not have that characteristics, is not considered a stem cells, and it can lose this from shear stress. Shear stress also can cause low viability. Okay, and it also can cause stem cell, especially to attain senescence early. So that means they can no longer propagate. So you cannot culture them and expand and scale them up. So there are many studies that actually talk about the connection between shear stress and this implication, losing its characteristics and attaining senescence early as well. So I have a few papers here that talk about that. And also, there are also studies that connect shear stress with the ability for stem cells to differentiate. So because the stem cells are meant to differentiate, when they have a lot of shear stress, they also, because they lose the characteristics and low viability and senescence, so they're not able to differentiate properly into their different cell lines as well. So this is a very brief overview of what shear stress is, and it's more prominent in stem cell because of their sensitivity, and later they have to differentiate, so they sometimes lose that ability from shear stress. Okay, so we talked a lot about what is the challenges. So now I give you solutions. So these solutions won't 100% eliminate the problem, but it will significantly improve the two challenges that we discussed, which is low yield and shear stress. The first is the use of bioreactors to increase cell culture yield. So bioreactors gives you the ability to culture your cells. So you put your cells in a T flask first, and then you increase the yield by using a bioreactor. So when you culture in bioreactor, you're no longer culturing in 2D flasks. When you use T-flask, the cells are all, so MSC is an attachment cell, so attached at the bottom of the flask. In the bioreactor, they become a 3D culture. So they use what we call a microcarrier. It's like a very small microscopic beads. So it creates a surface area for the cell to attach. So this 3D culture actually can encourage, increase the cell yield. And in the bioreactor, we have what we call propeller. So something like a stirrer. So the stirrer, when it is continuously stirred, it actually can increase the homogeneity of the temperature, the pH, the oxygen and the gases dissolve, and it also help to increase the yield. And bioreactors are what we call a closed system. So because they're closed system, they're not exposed to the air so much as the T-flask, because every time you manipulate the T-flask, you need to open the cover and close the cover. But in bioreactors, you don't have to do that. And you can monitor what is the pH, what is the temperature in the media itself using this monitor. And you can control it as well. Okay, if the pH is too high, you can actually add acid or base. And if the oxygen level is high, low, you can actually change it, and temperature you can control. So you can get more reproducible cell culture conditions, and they can easily be scaled up. So in terms of scaling up, we do have many options in Appendorf for the bioreactors. We have very small options if you're working in R&D, small volumes, 250 mils, all the way up to very big volumes, even up to 50 mil volumes, 50 liter, volumes. So how does it happen? How do you culture from a T-flask to a bioreactor? It's actually pretty straightforward. So I have it in a few quick steps, 4 steps. And this is a screenshot from a white paper that is produced by Appendorf. So actually, you can find this white paper online or in Appendorf websites, and you can actually just download and read this white paper in detail. So how we can culture in bioreactors is when you get your primary culture, it is still advised that you start with the T-flask because the cell volume most probably will be small. And then after that, you introduce the cells into a bottle. This bottle will have the microcarriers. So the cells will now attach to the microcarriers. And then once they are conditioned, we call it, so they're conditioned, acclimatized, and they're happy with the microcarriers, then we introduce the bioreactor. So we add them into the bioreactor. And of course, you can change media every few days and monitor them. And also from a small volume, 1 liter, you can increase to larger sizes of bioreactor. So it's pretty straightforward. The only media step is conditioning with the microcarriers, so they can attach the microcarriers instead. And in Appendorf, like I said, we have, this is just an example, but we have bioreactors for a small volume, large volume. So even if you're trying to scale up to preclinical or clinical trials, we do have that volume capacity for that 50 liters of culture as well. We even have production volume, which is 500 liters. I think that is for therapeutics total production for customer usage already. That is also available. And this will help to increase your cell culture yield as well. Okay, so that was Appendorf sharing. So I would like to show you some evidence. I have just found online a few papers that actually also talked about using a bioreactor for mesenchymal cell culture, and this helps them to increase the cell yield. And they talked very detailed about it. So I have 2 publications from Elsevier actually, and they used bioreactors, and I'm happy to report they also used Appendorf bioreactors. So this is just a few examples. You would most probably be able to find more if you were to search for it as well. Okay, so in order to overcome low yield, you look into bioreactors. That would be perhaps the first something that you can look into. And second one, the shear stress that we talk about. So shear stress is from the process that the cell goes through. So you cannot avoid that. You have to pipette the cells, you have to centrifuge the cells, you have to mince the cells. So how can we reduce shear stress? So our goal is to reduce, and we actually can reduce shear stress by using electronic pipetting tools instead of manual. because electronic pipetting tools, for example, the Appendorf Explorer Plus, and any pipetting tools, actually, you have the motor and you can set the speed. So you actually can set a very low speed. So usually the minimum speed is 1, maximum is 8. So 8, you pipette very fast, aspirate, dispense fast. 1 is slow. So if you set the speed very low, then you actually have low stress. The cells undergo a lower stress, a more calmer environment. And very importantly also, you can actually reproduce your speed settings. So batch 1 of stem cells, batch 2, batch 3, batch 4, if you use the same speed settings, there's no variability in your parameters. So the shear stress amount applied can be exactly the same. So your downstream experiment will be more consistent and reproducible as well. And if you have a larger scale practice, if you're looking at therapeutics, you can actually opt for completely automated liquid handling systems, which Appendorf also carries, like the EP Motion. And this will actually do pipetting for you, moving the cells, changing reagent. So actually, it's a completely automated system. And actually, it has even lower, it will introduce even lower shear stress to the cells. This is something you can consider. So when it comes to electronic pipettes, the Appendorf electronic pipette was given the gold seal of quality in 2021, and that is given only to about 0.1% of all global lab products. And we are very, we really look into something we call physio-care concept in Appendorf products, not just for the pipette, but for everything. So if you use the pipette very often, you're probably very familiar with this movement. You have to pipette up and down with your thumb. Yeah. So after a while, you start to feel pain here in your wrist. Maybe if you start to feel pain here on your wrist. If you are new mothers, maybe you're also familiar with that. Sometimes new mothers also have that. We call it mother's thumb, and that can be induced when pipetting up and down. Imagine many years you pipette like this so many times every day. So now you're young. Now we are young. Hopefully, then later when you're older, then you may have what we call RSI, which is repetitive strain injury, and it can lead to things like nerve damage and also carpal tunnel syndrome. Yeah, so there's a nerve damage here, and we want to reduce this. So actually, when you use the electronic pipette, you don't have the up and down motion with your thumb anymore. So you actually reduce this in the long run, so you take care of yourself better. And also we have what we call the spring-loaded tip cone. So if you're familiar with the old pipettes, usually what you do is when you want to attach a tip, anyone here who will bang very hard on the tip until really it attach. So actually that is stressful for your hand and also stressful for the pipette, but and also stressful for your tip. But we can reduce that using the spring-loaded tip cone. So you don't have to do so hard. You just have to press down until the spring. There's a haptic feedback and you can attach. So it's all tested to make sure that this haptic feedback is a very consistent attachment, reproducible, and gives you great pipetting as well. Okay. And not just pipetting, because shear stress can also be introduced by centrifugation, right? So we want to make sure the centrifugation is suitable for your stem cells. So you have to choose the right centrifuge with the right rotor, and very importantly, the right adapter. So not just Appendorf centrifuge, but any centrifuge that you choose, these are important criteria to look into. But in Appendorf, we do have very versatile adapter option. So for example, if you see this GIF, so you can see that it keeps changing, but the rotor and the bucket is the same. Only the adapter inside the color ones, they keep changing. And this is what we call a universal rotor and universal adapter. Universal because one adapter, you use the same rotor, you use the same bucket, and you just change the adapter. And even then, one adapter can take, for example, the yellow one can take a 15 mil tube, a 5 mil tube, and a plate, a deep well plate as well. And for example, the blue one can take 25 mil tube, 50 mil tube, and plate as well. So you don't have to buy different types of rotors and adapters to fit the different vessels. So if many people in your lab, some use plate, some use tube, we can just use the same adapter. But choosing the right adapter is very important. because it will help you to lower vibration. So every time when the centrifuge spins, the centrifugal force will actually cause it to have some vibration. So we want to reduce the vibration because the vibration can cause shear stress to the cells. So choosing the right centrifuge that has a good stability and low vibration can help you to reduce the shear stress for your cells. Okay, so that was only the first part, which is sample collection. So how can we reduce shear stress? By increasing yield using bioreactors and choosing the right tools, pipetting and centrifugation tools. Now we look into the second part. You have already processed your samples, so now you have to culture them in the incubator and check them every day. So what is the challenges that you face at this step? So maybe this step is the challenge where many people can relate to if you work in the cell culture lab. So of course, the first one is low viability, a poor cell growth. Sometimes after a few passage, you cannot grow them. It could also be because they attain senescence from the shear stress, and also poor cell morphology and differentiation and characteristics. So when we talked about the different characteristics that make an MSC, so they have to have certain kind of surface markers. For example, they have to have these surface markers. They shouldn't have these surface markers. But sometimes when you check and you see sometimes your MSC have no CD73, that means it is no longer MSC anymore. Yeah. And last but not least, but very big issue, I think, is contamination. Because especially if you're doing primary cell culture, like for example, the umbilical cord, it is obtained from the baby at full-term delivery in the operating theater. The operating theater is a clean room, but it's not exactly sterile. And then when you bring it back and you try to culture in the, it's very difficult to maintain this, very difficult to make sure there's no contamination, especially because you cannot use antibiotic in the media. So sometimes when we do normal cell culture, it is quite common practice to add antibiotic to ensure there's no contamination, but you cannot do that for stem cell culture. So contamination becomes a bigger issue here. And like I said, there's a lot of implication online that you can find on studies that talk about how having different characteristics, different morphology is can affect stem cells and their differentiation. I have a few papers here. I also have a paper that talks about hypoxia and its effect on stem cells. So hypoxia is low oxygen concentration. So growing in low oxygen concentration. So maybe now you're wondering, what am I talking about? Low oxygen, right? So I have a question from you. So this one I want to hear from you. You can tell me again in the chat box. Yes, no, not sure is also okay. You can tell me if you think oxygen control is crucial for stem cell culture.
Yes.
Okay. Okay. So I see many yes.
Yes, so many yes.
Okay, so that's nice. Okay, so I only see yes. Okay, that's good. That's nice. So maybe I can keep my next slide.
No, from
Okay.
Badila.
Okay. That's good. So we can all find out together. So you can also check with the colleagues who said yes for more details later. Okay, so many yes. So many yes. So you think it's yes. So I'm happy to say that you are right. Oxygen is actually very, very important for stem cells. And why is that? So I want you to think about the most stem cell, how to say, like the most stem cell. So the cell that is most stem cell is actually the zygote. So the zygote is the embryonic stem cell that it can become any kind of stem cell. It is totipotent, it's a class totipotent. So this zygote, when it forms during fertilization of egg and sperm, and it's only a single cell, and it's found inside the mother's womb. And for this cell to receive oxygen, because it's one cell and it's inside the womb, so it's very deep in. So imagine for this cell to receive oxygen. Do you think it will be easy or will it be hard? So you can also tell me in the chat box. So no question here, but you can just tell me yes or no. Do you think that the cell can? Okay, so I see.
Answer.
Okay, so yes, actually it's a little bit difficult for the zygote, the most stem cell, to receive oxygen. But that being said, the fetal maternal oxygen blood exchange is very, very fantastic. I think it is a biological miracle, but generally it's difficult for the cell to receive oxygen. So the mole stem cell is growing and can become all the kinds of stem cell in a low oxygen environment. So what is the oxygen environment that it becomes all these different types of cell? So actually the different types of stem cell grow better at different oxygen concentration and low oxygen concentration. So for example, mesenchymal stem cell, they actually require only 2 to 8% of oxygen to ensure that they're growing very well and to maintain their mesenchymal stem cell characteristics. And neural stem cell, sometimes even less than 1%, they can still grow because the zygote at the beginning, when it's forming its brain, which is like fully formed by 4 weeks of gestation, they can still grow even at 1% oxygen. And for example, hematopoietic stem cell only require between 1 to 6% of oxygen to be hematopoietic stem cell. So very low oxygen concentration. So that being said, I always like to ask this question. You can answer me. No correct, wrong answer, don't worry. I'm just curious. So the stem cell, requires 2 to 8%, 1 to 8% oxygen. What about the normal cell? When we culture normal cells or when we're breathing our normal cells, what is the oxygen requirement for it? So not stem cell, the normal cells. Like when you culture in the incubator, does anyone know? You can share in the chat box.
How guys?
How many percent of oxygen do you think the cells need? 21. Wow, okay.
30%.
Okay, that's nice. Okay, so okay, so I see more 20, 30. That's very nice. And you are absolutely right, actually, because the oxygen concentration in our atmosphere is about 20%, 20, 21 plus minus, and the rest is nitrogen and a little bit of other gases. So actually when we breathe in, so maybe our cells don't receive exactly 20%, but plus minus, they actually require about 20%. But the stem cells require lower than that. So that's why oxygen control is very important. Okay, so that means when you choose the incubator, you have to choose the right incubator, one that can give you safe cell growth in a good oxygen control environment and also prevent contamination. So choosing the right incubator is very crucial. So the first thing this incubator must have is O2 option. So it must have oxygen option for stem cells. It must have oxygen control for stem cells so that you can control the oxygen, can reduce it to ensure that the cells are maintain their stemness. And then you want to choose an incubator that is fanless. So sometimes you choose you see an incubator, and inside the incubator there's a fan. If you are familiar, there'll be a fan. So why do I say fanless? It's because fan, meaning the fan is turning, that means there's turbulence and vibration. So this turbulence and vibration can cause shear stress, especially for your stem cells. So we don't want to put them in an environment where, as they're trying to grow, they constantly have shear stress. Yeah, so that is quite bad for them. And of course, you choose an incubator that can give you fast temperature recovery. So ideally, your CO2, because every time you open the door, the CO2 concentration is going to disperse. And when you close it, then it will recover back. So there is a gap there. So we want to make sure that as it cannot be avoided, any brand, any type of CO2 incubator that will still happen. But you want to choose one that can, when you close the door, the CO2 and the O2 recovers quickly back to the set temperature, ideally within less than 5 minutes. So it doesn't affect your cells too much because stem cells are very sensitive. Yeah. So, and of course you want incubator that can allow you to have contamination-free features. So for example, for the Cell Expert C170i from Appendorf, we do have what we call a HTD. So this is a high temperature disinfection. So it can actually disinfect the entire incubator at 180 degrees Celsius. And we advise to do this every month, at least. but that is plus minus there. But at least you have the ability to decontaminate your incubator itself. And of course, we also advise that you must clean your incubator. So any kind of incubator, you should actually remove all the parts, remove the racks, and you should actually clean them with disinfectant, preferably 70% ethanol. So for that, this process, it's made easier if you have a very easy to clean incubator, so that it's not such a big task, because nobody wants to do that, because nobody wants to clean the incubator. We always fight in the lab. Okay, your week, your turn, my turn. So if it's easy to do, then it's easy for everyone to do. You don't feel it's such a burden, So we do have smooth, seamless chamber to ensure that you can wipe everything easily. We don't want any areas where there is like gaps inside, or metal areas where behind is very hard to reach. If it's very hard to reach, you're most probably not going to wipe, and that can cause bacteria to grow in that area as well. So this actually helps you to keep your cells contamination free. And like I said just now, the temperature is temperature recovery. When every time you open and close the door is very important. And also temperature homogeneity to ensure reproducibility is very important. So I have here, this is also from a white paper from Appendorf. You can Google it. On a website, you can also find it. You can download and read that. So it's we are looking at what is the temperature inside the incubator compared with Appendorf C170, compared to a few different competitors, different brands. And we have this graph. So the ideal range is between here. So I assume around here is the 37 degrees, which is normally we set at 37 degrees. So plus minus a little bit, still okay. But if it's too red, that means it's very high, maybe 39, 40, I'm not sure. If it's too low, perhaps it's about 34, 35. So there's a big difference. So the ideal range is 37. It is very, very difficult to maintain exactly 37 at all times in all the place. So because you also open, close the door. But what we want is we want to make sure as close to 37 as possible, so that the cells are reproducible, they maintain their stemness. Okay, so when we checked it, so we did 27 probes and checked at all the different location here. So every ball is, imagine is a probe, checking the temperature. So when you see the temperature here, they're all about bluish green. So they're somewhere in between this range. As you can see, even the Appendorf one is not 100% all green. So there are some little bit warmer spots, because it could happen. That maybe is recovering a little bit sometimes. But generally they are homogeneous. But if you compare, some other incubators can have very varied temperature homogeneity. For example, this one, so at the bottom shelf is all blue. So that's about 34, 35 degrees. The top shelf is very warm, and the middle shelf is very ideal. So when you have this kind of incubator, or even this kind of incubator, what happens is, if today put my cells in this shelf, they grow very, very well. I managed to passage many times, very good viability. Tomorrow, I get a new batch of samples and I put them here. And after 2 passage, maybe they stop growing, they have senescence, and I cannot get good viability. And maybe 3rd time when you put it here, you also find the same issue. And you may be wondering, what is the issue? Why my cells cannot grow? And maybe it never crossed your mind that that is because of where you put them in the incubator. So it's very hard to say. Without testing and service IQ, OQ installation and certification, temperature mapping, it's very hard for you to know where is the place you should put your cells. So you want to choose an incubator where it doesn't matter. I put here, I put here, I put here, today, tomorrow, at least they are more reproducible and almost the same situation. They are more same. So that is the kind of incubator that you want to choose. So how can Appendorf achieve this? How can we ensure that they are all almost 37? It's because we have heating in all sides. So we have all this red color thing is a heating coil. So this is inside the incubator. So there's one at the back, and there's a one like this from the top to the sides. There's one at the bottom, And there's one from the door, because the front one is a door, right? Open, close. There's one coming from the door as well. So the heating is in all sides. So it creates a good convection to ensure the temperature homogeneity is very good. So when you see results like this, most probably the heating is focused on the top, and there's no heating coil at the bottom. So that's why you see results like this. And if you see like bottom, then maybe the heating is more focused at the bottom. Or for this one, the heating is most probably focused at the back. Okay, I'm not 100% sure because I'm not sure which competitor this is, but when you look at the pattern, the heating coil may be not in all direction. So you want one that can give you homogeneous temperature. Okay, and how can you avoid contamination? So I don't have to go so much detail into this. I think you're very familiar with contamination in cell culture, especially MSc culture. So there are many papers in there. But what I want to hear from you that do you face contamination issue when you're doing cell culture or stem cell culture? You can say yes, no, just curious.
Yes or no, guys. Type in comment section.
Yes, okay.
Yes.
Okay, thank you for sharing honestly.
Yes, yeah. So many yes.
Okay, thank you for sharing honestly. Personally, me as well. I think when I work in the stem cell lab, there are many contamination issues. So sometimes we don't know where it's coming from. So it's quite common. I think from time to time there may be contamination issue in the cell culture. So it's very common. So how can we overcome this? I think that's very important. Okay, Okay, so how can you overcome this? So contamination can happen from many things. So the first thing I want to talk about is aerosol contamination. So I think you're very familiar with aerosols. So because of COVID, aerosol is very common. So you sometimes when you talk, you generate aerosols. So the COVID virus, for example, is actually inside your aerosols. So actually any activity that involves any form of liquid sample and any kind of movement actually can create aerosols. So even pipetting up and down can cause aerosols. Even pouring your liquid, it can cause aerosols. Opening your culture can cause aerosol. Shaking your machine can cause aerosols. So how can you prevent aerosols? Actually, you cannot really prevent aerosols. All you can make sure is that you use the right equipment to ensure that there's no contamination from aerosols. So when you are pipetting, you actually create aerosols. So I would like to suggest the usage of filter tips in order to avoid cross-contamination. So how does cross-contamination happen using aerosols? So this is a picture of a pipette, and this is the pipette tip. So let's say you have your sample 1, umbilical cord stem cells from a patient, but just assuming. So you aspirate this sample, and then when you aspirate, you create aerosols. So aerosols go inside your pipette tip, and they go inside the pipette nose cone as well. So it's this area here. They actually can go there. And then in this patient, just as an example, is, for example, COVID positive. Okay, this sample is COVID positive. And then you follow the correct procedure, you discard this tip, and you take a new tip. So that is a correct procedure. And then you use for sample 2. So sample 2 is COVID negative. And when you aspirate, you create aerosols. And then when you dispense sample 2 inside the tube to do downstream processing, aerosols from sample 1 also go inside the sample. And it's possible that when you get the result, both sample 1 and sample 2 can read positive for COVID. Yeah? So this also is the same way for contamination. For example, sample 1 is contaminated with bacteria, and bacteria is in the aerosol. And when you aspirate sample 2, it's in your culture as well. So not just your sample. So let's say everybody is sharing the same pipette in the biosafety cabinet. So person number 1 in the morning has a contamination. That means that everybody who use it and for the next 2-3 days may have contamination, but they don't realize it because it's inside the nose cone here. It's possible. So because we share the same pipette. So how can you prevent this? Is by using what we call a dual filter tip. So this dual filter tip, actually, you can see there's a filter here. So when you aspirate, aerosol is still generated, but it's captured in the filter. So Appendorf has a dual filter layer. They have different sizes in the pores. So this captures larger sizes, this captures smaller sizes. And the Appendorf dual filter is actually certified ISO class and has a 99.5 HEPA class filter efficiency. So we do have certification for it. So it can even retain microscopic contamination and even viral contamination as well. So just by using filter tip, you actually can eliminate cross-contamination, especially during MSc culture. So you can also choose the correct grade. So we have tips in different grades. So you choose the tips that is sterile grade. So they are sterilized when you purchase them. So they have you can actually reduce contamination as well. And like I said, everything generates aerosol. So not just pipetting, centrifugation also generates aerosol. So I have a video here. Let me see if it plays. Okay, so this video has no audio, so I will narrate for you. So here you can see that you're centrifuging your sample. Okay, it's contaminated and there's a breakage in the tube. There's a sample leakage. So your sample is going to leak into your chamber, and then when you open the lid, it's going to leak into your chamber. Okay, so when the next person use it, they're also going to cause cross-contamination. So how can you avoid this? Is using something we call aerosol type lids. So Appendorf centrifuges, most of it, I think almost all of it, all the rotos comes with what we call as aerosol type lids. So they don't allow aerosol to escape in the roto. So let's say there's a centrifugation, there's a breakage of the tube. If you want to ensure safe centrifugation, don't open the lid in the centrifuge, remove the whole rotor and open it inside the biosafety cabinet. So that in that way, you actually can completely eliminate cross-contamination. And not just for fixed angle rotor, even swing bucket rotors, we have aerosol tight lids. And these aerosol tight lids are certified by third party. So it's not certified by Appendorf. They are certified by the Health Protection Agency of England. So whenever let's say you buy a centrifuge, you'll receive a certification in the manual. At the back of the manual, there will be their certification. It will tell you the centrifuge that you bought, the rotor, and they tested each centrifuge rotor specific. So they tell you rotor by rotor whether this is whether it has a certificate for containment testing or not. And you will get a certificate for all the different types of rotor, and it's a containment testing for aerosol safety. Okay, so using the right centrifuge, using the right filter tip, and this can using the right incubator can help you make sure that your cell culture is, especially stem cell culture, is reproducible for a longer time and no contamination. And then the last step, storage. So many people actually don't think about storage steps so much. They don't consider that so much. But I'm here to tell you that actually, because stem cells are very sensitive, the temperature fluctuation, even in the short time in the storage, actually can cause the cell to lose its characteristics, viability, and functionality. So what a person may experience is they have propagated the stem cells, they have passaged it, and then now they're not doing the downstream experiment yet. They are storing first. So they're going to collect all the stem cells before they do the downstream experiment, for example, and they store it in -80 for a short while, maybe 1 or 2 weeks, maybe 3 weeks, depending. And then when they remove it to actually do the downstream experiment, they find out that the cells not going to not propagating, or they don't have the surface markers anymore. They consider them as stem cells. And why is that? It's because of temperature fluctuations. So you may also Google it. So there are some papers that talk about small temperature fluctuation and how it can affect the parameters and the differentiation potential, especially for stem cells. So even transient warming, even changes between 2 to 4 degrees for 2 hours can negatively impact stem cells. So very small temperature difference and very short while, because they're so sensitive. So you choose, so especially for short time storage, it's very important to choose a freezer, like ultra low temperature freezer that has good temperature stability. And how do you ensure you have a good temperature stability freezer? You have to choose freezers that has characteristics that can ensure better temperature. For example, freezers that have vacuum insulation panels, and also freezers that has a flat gasket to ensure it has a better what we call a warm-up time. So what's a warm-up time? So warm-up time means the time it takes for your freezer to go from -80 to -50 in terms of no power failure, for example. Let's say you have a power failure, and so how long it takes to go here for this value. So the longer it takes, it gives two things. It means two things. One, it gives you time to troubleshoot that issue, the power failure issue. Second, it also means that the insulation quality of the freezer is very, very good. So there will not be temperature fluctuations between 2 to 4 degrees in a short time as well. So this data you should be able to find online. For Eppendorf, we have technical data for all the different types of freezers. And the other brands as well, you can find them online. They will tell you what is the warm-up time, what is the time it takes for the temperature to go from -80 to -50. So what we want is something very long. So that gives us the confidence that we have very good insulation quality and lower temperature fluctuation. So if we compare, for example, the Eppendorf 570, the warm-up time is 11 hours. And here is less than half of that. So that should give you an idea of the quality of the insulation. So you want to make sure that your sample is safe even during power failures and there's no temperature fluctuation. And this one, the warm-up time and the insulation quality will also give you an idea. Every time you open and close the door of the freezer, there's temperature fluctuation. So the most important thing is when you close the door, you want the temperature to go back to -80 very fast. So if you fluctuate too much between 2 to 4 degrees for 2 hours, then it can negatively impact your stem cells. So I do have a comparison here. So you want to make sure that you choose the freezer that gives you faster door recovery time. So every time you close, very fast, it can go back to the -80 temperature. So for example, in this brand X, where they set to -75 degrees, and it takes them 24 minutes after they open the door for 1 minute. When they close back, it takes 24 minutes to go back to -75. So if I compare that to the Eppendorf 740, so we set to -80 because that is more common than -75. And we give detailed information on the different times. So not just 1 minute, but if you open for 15 minutes, if you open for 30 seconds, if you open for 120 seconds, how long it takes. So if you see here, if you compare 1 minute, which is 60 seconds, to 1 minute here it takes 27 minutes. So you may think that's longer, but it takes 27 minutes to go back to -80 compared to 24 minutes to go back to -75. So most probably if you go back to -80, it's going to take a little bit longer than that. So that's why the data is shown at -75 instead of -80, so that the information looks more acceptable generally. So these are things that you can look up online to find this information. Okay, so that is the first part on stem cells. I do want to talk a little bit on exosomes. So this is the same picture, history chart on stem cells. So now suddenly we have something called stem cell derived exosomes. So I think I'm a little bit over time. Is it okay if I take another 5-10 minutes?
No problem, Roshini.
Okay, all right. Sorry for that. So maybe we started a little bit later. Okay, so I'll just talk a little bit on exosomes. So stem cell derived exosomes. So it's very, very popular now. I think in the last couple of years, it has been gaining more popularity. Okay, so why is that? Why is stem cell derived exosomes so popular now? Because they have discovered something. So I want to ask you, can stem cell derived exosome execute stem cell benefits without using stem cell itself? So what do you think? Can you substitute stem cells for the exosomes derived from stem cells? So no right,
Yes.
wrong answer.
Yes.
Okay.
Yes.
Yes, it's true. So you're absolutely right. So I see all the answer is yes. That is exactly the reason why stem cell derived exosomes is very popular now. So exosomes is basically an extracellular vesicle that is secreted by the stem cells, and their main function is cell-to-cell communication. So they carry biomaterials like DNA, RNA, protein, biomarkers that can actually communicate to another cell what you should do now. So they actually can communicate cell differentiation as well. So you theoretically can harness the potential of stem cells using the exosomes without using physical stem cells. So physical stem cells sometimes have some bioimmunity compatibility rejection. So because they are physical cells, sometimes the host rejects that cells. So you actually can reduce that using exosomes. And because exosomes are so small, they can even cross the blood-brain barrier, which stem cells, physical stem cells cannot do. So now you can target even malignant tumors in the brain as well, and have the same benefit as stem cells. So that's why it's so popular. It's still slightly, we are still working on understanding stem cell derived exosome better. I think it's really gaining momentum. Okay, so how much momentum is similar? I have here data that shows that the exosome market will surpass more than 2.2 billion by 2030 as well. So a lot of momentum for exosomes and stem cell derived exosomes as well. So if you want to know what are the different pharmas that are currently working on exosome, especially in the Asia Pacific region, so more relevant to us. So you see there's many here, and I'm very sure that you're quite familiar with a few of the big name pharmas here. So they are all working on either drug development or even clinical trials on exosome as well. So it is something that is really gaining momentum. And when it comes to exosome, so it's the same thing. You do the same workflow. You must culture your stem cells, and this exosome is secreted in the cell culture media. So the exosome is here because it's secreted by the stem cells. I think the biggest challenge in stem cell derived exosome is trying to isolate the exosomes because they're very small. So first thing you need to do is you want to remove the stem cells. So you have to remove the cell and cell debris. That's very simple. You do centrifugation at low speed, remove the cells, and then you can do some differential centrifugation to remove maybe cell debris and the different smaller components of the cell. But to isolate exosomes, you need the speed of ultracentrifugation, which is 100,000, at least 100,000 to 120,000 G, or even 180,000 G. I think even 200,000 G some people use. So this speed is very, very high, and that means it can have a lot of challenge, very time consuming, very low yield process. So how can you overcome it? Is by choosing an ultracentrifuge. So first you need an ultracentrifuge, but we want to choose an ultracentrifuge that makes your everyday process easier. So the Appendorf ultracentrifuge, we have a few models. You can come back to us if you want to know more about those models. But what I want to talk to you about is one of its ability. So if you're familiar, if you've done ultracentrifugation before, so every time you're doing a centrifugation, so normally normal centrifuge, any centrifuge, you must balance. So everything that happens on one side of the rotor must happen on the other side. So balance them. Very simple. In ultracentrifuge, this balancing is even more crucial because it's going at very, very fast. 100,000 G, 200,000 G. So how we balance is we balance using a weighing balance. So you cannot just put 5 mil here, 5 mil here, and assume it's balanced. You have to take the tube physically and put in the weighing balance. So if you have a fixed angle rotor, this tube must be balanced here, this must be balanced here, this must be balanced here, and this must be balanced here, so on and so forth. So, and how much is the difference? The allowable difference is only 0.1 gram, not more than that, because of the speed the ultracentrifuge is going. So I have worked with ultracentrifuge before, and we worked with viral samples. So I can tell you it's very, very personal experience. It's very, very tedious because the viral sample, we must manipulate it only in the biosafety cabinet, but the weighing balance and the ultracentrifuge is outside. So I have to physically walk to the weighing balance, and then I check. If it's not within 0.1 gram, I have to go back to the biosafety cabinet, add some more virus sample or dilute it or remove some sample, and then do this back and forth for all my tubes, make sure they're all balanced before I can even run. So sometimes it can take up to 20 minutes. It really depends. So it can be it can be very time consuming. So already you have to have an ultracentrifuge, and already the exosomes from stem cells very low yield, and now it's so time consuming as well. So we want to reduce some of these challenges. So what you can, if you choose the right ultracentrifuge, for example, the Eppendorf versions, these are all the ultracentrifuge we have. We have what we call visual balancing. So meaning you don't need to use weighing balance. You actually can just use your eyes visually. Just make sure that the liquid amount here and in the other tube is not more than 5 ml. Then it's actually fine to just centrifuge it. If let's say it was more than 5 ml, but you mistakenly thought it's 5 ml, you don't have to worry, because the sensor will still pick it up and tell you that it's imbalanced. But you don't have to use any weighing balance. So this can really save up time, even 10 to 25 minutes of the sample time as well. And if you are very familiar, when you use exosome as well, all of this process, when you collect the sample, supernatant from the stem cell, you're centrifuging it, and then downstream, all of this sample, you need to put the exosome, it will be in a solution, you need to put it in a tube. So choosing the right tube is also very, very important. So I would like to introduce you to the protein low bind tube. Oh, actually there's a video here. So you can hear some background sound. Can you hear that?
Yes.
Okay. Okay, so I would like to introduce you to the protein low bind tube. So Eppendorf has two different types of low bind tube. One is DNA, one is protein. And they are made primarily with the idea, because normally protein or DNA samples, they have adhesive effect, hydrophobic effect, and they actually will adhere to the tube wall. So as soon as you put your sample inside, and when you aspirate them out, right, you find that the concentration of DNA can be much lower, for example, DNA, than the amount you put in. And that's because some of the DNA already adhere to the side of the wall, and they're not able to, you cannot recover them anymore. So we have the protein and low bind tube meant for protein and DNA samples. But there's studies online. So this is not a study done by Eppendorf. This was discovered by Eppendorf, I think. It's a study done third party in a PLOS publication. So they actually used the Eppendorf protein low bind tube to test it whether it can help to reduce loss of exosome, because exosome is very low yield. So they actually used the Eppendorf tube. So they actually I have a graph taken out from this paper. They used the Eppendorf tube compared to the Eppendorf protein low bind tube, and they also compared with some coated tube, and they compare it with a different brand, and also the different brands maximum recovery tube. So the maximum recovery tube has the ability to also reduce surface binding and recover. So it should have the same functionality as the Eppendorf low bind tube. But we can see that from this graph, when they use the Eppendorf low bind tube, they're able to recover 80% of the sample compared to when they use a standard tube. So if they put in 100% sample, after 48 hours, they're able to recover 80% using low bind, and they lose almost 60%, and they don't use low-bind. They only can recover about 40%. And if we compare with other brand as well, you can see that even the maximum recovery tube is only recovering about less than 50%. You know, it has no much difference in terms of recovery compared to the standard tube as well. So this is a great addition in your exosome research. So we would recommend to use the protein low-bind tube. And it's been, this is one of the first few papers, but I think there's more than one now that talks about how you can actually utilize the protein low-bind tube to increase sample recovery so that you reduce the adhesion of your exosome to the tube during your exosome research. So when you collect your sample, when you store your sample, it is recommended that you choose the right tube as well, because we also have a lot of evidence that even the tube can cause you to have low yield because the samples all adhere to the tube surface. So the appended protein low-bind tube has no coating, so it's just it's manipulated at a molecular level to achieve this low-bind effect. Okay. And that would bring me to my very last slide. So there are many challenges in stem cell culture and stem cell derived exosome culture. And we don't have 100% solution for any of it. But we do have solution that can actually give you very good and very significant improvement. And that is the choice of using bioreactors instead of T+, electronic pipettes, choosing the right centrifuge and the roto, and choosing the right adapter, very important, reduce vibration, choosing an incubator that has oxygen control and a good quality freezer, and last but not least, an ultracentrifuge if you have exosome, if you want to isolate exosome, that can make your process a little bit easier, and to consider to use the protein low-bind tubes throughout your research with exosomes as well. That will be my last slide. So before I move on to the next one, I'm open for any questions, if you have any questions.
Yeah, okay, thanks, Rusiani, for your great explanation and also comprehensive information, yeah, especially mesenchymal stem cell, and also you tell us about the market potential, challenges and solution. Also, we can hear about the shear stress and yeah, the low characteristic, low viability, and also the solution with the culture in bioreactor, and we can explain about the choosing the right tools, pipetting, centrifugation, hypoxia, oxygen control, and also storage also in ultra freezer. And the last but not least is stem cell derived exosome, ultra centrifugation, and also choose the correct tube. So back to the participants, any question to Roshani? Please feel free to unmute yourself or type your question in the chat box. Ada pertanyaan Bapak Ibu? Mungkin bahasa Indonesia juga nggak apa-apa.
Ya, nanti diterjemahkan sama Pak Hendy.
Iya, betul.
Oke ya.
Silakan, Bapak Ibu.
Monggo Bapak Ibu yang ingin bertanya langsung boleh. Oh, ada ini Pak dari Evi Istiqam. Oh, bukan. Oh ya, yes.
Yeah.
The question.
There is a question from Ibu Evi Istiqomah. Yeah. Thank you for your presentation, Ms. Ruseni. I want to ask about the exosome content. Are the growth factor contained in exosome?
Okay, so growth factor. So I'm not sure if I understand the question correctly. Yeah. But usually growth factors are used for stem cell culture, and they need to be used as well. And the exosomes, if you are requiring exosome from certain type of stem cells, then you need to culture with growth factor as well. But growth factor itself is your exosome can contain protein molecules as well in terms of communication, but it depends on this exosome is derived from where. Yeah. So there's a big variability in that, but you still need to culture the stem cells with the correct growth factors. Yeah.
Yeah. Okay. I think Roshani answer Ibu siapa tadi? Ibu. Yeah.
Ibu Evi.
Ibu Evi. Okay. And then I'll move to the next question, Ibu Fatia. Hello, I want to ask about targeted protein that could be isolated from exosome. How can we isolate the certain protein from the whole exosomal protein if that targeted protein via novel and never be isolated before?
Wow, it's a very challenging question since it's novel and never been isolated before. But so this is a very difficult answer. So my honest answer is with a lot of trial and error. So a lot of experimentation and optimization. But I think the most important thing is finding what is the size of the protein that you want to isolate. And you can use filtration methods to eliminate the different types of proteins. So you have to isolate the exosomes first. And then after that, you need to maybe isolate the different types of components, and you can actually exclude them based on their size. And if you know the size of your protein, then when you choose the right speed of the ultracentrifuge for the size, and also maybe couple it with some filtration, molecular weight cutout filtration membranes, then you may be able to isolate your novel protein, but it may require a lot of trial and error.
Thank you, Rosani. Ya itu ya Bu Fatia ya. Jadi perlu banyak trial dulu ya, karena memang yang itu kan novel dan belum pernah dicoba juga. Jadi dicari dulu untuk size-nya, nanti untuk speed atau sentrifikasinya bisa menyesuaikan. Tapi kalau memang pengen tahu identifikasi proteinnya itu apa ya, harus pakai mass spectrometry ya. Jadi nanti ada beberapa workflow yang memang ditambahkan di situ.
Yeah.
Okay, maybe the last question from Dolly Wong. Yeah. Have you worked with stem cell from insects such as silkworm?
Okay, sounds very, very interesting. But personally, I haven't worked with insect stem cells before. I've only worked with a human stem cells cultures before, actually. So because we were aiming for therapeutics, potentially therapeutic usage. But it does sound like a very interesting research area, academic area. Yeah, unfortunately, I have no experience in that.
Okay, yeah. Thank you, Miss Ruseni. And ya, mungkin saya akan kembalikan ke Mbak Aida dari Indonesia. Terima kasih atas pertanyaannya. Saya titip salam untuk Pak Didik juga, untuk Mbak Evi, Mbak Turi dan tim ya, untuk kesempatannya yang sampai mungkin ini pada ngantuk ya, karena udah siang agak sore, tapi kita bersama semangat ya untuk diskusi tentang stem cell di exosome itu. Nah, nanti Miss Ruseni kita akan terbang ke Surabaya sebenarnya tanggal 31 Oktober. Nanti di Hotel Bumi Surabaya, nanti kita bisa share untuk undangannya. Mungkin Ruseni ada could you please add something to the feedback or
Yes. I do have one final. Actually, I have two final. So feedback. So we normally after every talk we try to get your feedback so that we can always improve on this talk, and also to see which are the informations that we can add, delete, which did you find interesting. So I do have two feedback forms. I will be really grateful. I would appreciate it if you can fill it for me. So this one, if you can scan the QR code, is NPS form. So it's very quick, just 3 questions. If you enjoyed this talk, if you think it's beneficial and we should continue to do talks like this, please rate us a 9 or 10. And I do have another feedback form, which is more detailed and it will take a little bit more of your time. So maybe you can just screenshot it shortly and you can answer it in a while. So if you have the time to screenshot this, I will probably leave this for about 30 seconds. And then I'll change it to the other feedback form. So this one is very quick. The other one will give us more detail. So the feedback form is just to ensure that the content is appropriate, and so we can continue to do this kind of talks, and that it is something that it's useful for everybody. So if you have done this one, I will show you the other one. So this one is a little bit more in detail. So it allows me to get to know some things like what did you learn, and also what are the other future things that you would like to hear about from Appendorf as well. So two feedback form. One is a quick one.
Okay.
Quick one and a detailed one. Okay. So maybe I can also share the quick one on the chat.
Yes.
Just give me a minute. Okay, I'll share this in the chat, and I will leave the quick one on the screen.
Yes. Oke.
Oke.
Oke, ini untuk flyernya juga sudah di-share oleh Pak Hendy ya, Bapak Ibu ya, jika ada yang ingin join juga ya Pak Hendy ya.
Ya, tim
Nanti.
Indio silakan ke Surabaya juga. Ya,
Oke, siap Pak. Nanti kami bantu share juga. Oke, dari Pak Hendy ada yang ingin disampaikan Pak sebelum kita tutup?
Oh, sudah cukup. Terima kasih banyak atas kesempatannya. Thank you, Rushani, for your support and for your great information for us to do the stem cell, tackle the stem cell challenges. Yeah.
Okay, thank you so much. Thank you for staying back later than the scheduled time as well. Truly appreciate you staying back as well.
Yes.
Thank you so much. Yeah.
Oke.
Terima kasih.
Yeah, terima kasih. Okay, Ms. Rusini, thank you very much. Your presentation was very, very good, and thank you for Mr. Handy as moderator today. And okay, finally we come to the end part of our webinar, everyone. I would like to thank again our speaker for Ms. Rusini, your informative and interesting presentation. And Mr. Handy juga, terima kasih. Dan semoga kita bisa bertemu di next agenda. Oke, dan saya mewakili dari tim Inbio mengucapkan terima kasih juga jika ada kekurangan ataupun yang ini kurang dari saya penyampaiannya. Thanks again, Ms. Rusini. Okay, see you next again, hopefully. Before we close our webinar, please on camera, we will take one picture together with Ms. Rusini too.
Okay.
Okay, guys. Sudah siap? Miss Rusini, could you stop your
Okay.
screen
I'll
share?
stop
Yes.
sharing. Okay. Yeah.
Okay, guys. 1, 2, 3. Okay, one more. 1, 2, 3. Okay, guys. Thank you so much, Miss Rusini, Mr. Hadi, Pak Hendy. Semoga kita bertemu dengan waktu. Saya akhiri. Wassalamualaikum warahmatullahi wabarakatuh. Hopefully see you soon again, Miss Rusini. Thank you.
Okay, thank you. Bye.
Yes. Thank you.